Welcome everybody to today's Carney Conversation. I'm going to do a quick introduction to our guest today, and then we'll be kind of moving pretty quickly into kind of a question and answer discussion with Kate. And I just wanted to remind everybody that if you want to post questions, you can do that in the Q&A, and Chris, who I'll introduce in a second, and I will be taking a look at those and then posing those questions to Kate. So again, thanks for joining us for this Carney Conversation, CRISPR and the role of gene editing in neuroscience. And it's really my delight to welcome Kate O'Connor-Giles, Professor Giles. And Kate is the Provost Associate Professor of Brain Science, and she's also in the Department of Neuroscience here at Brown. And the other person that's here with me today is Chris Moore, who is also in the Department of Neuroscience and Associate Director of the Carney Institute and Professor of Neuroscience. So thanks for joining us, Kate. It's really a pleasure to have you here.
Thank you very much for having me.
So I'm just going to say just a super quick summary of your work, which it's not going to do it much justice, but I'll just get the ball rolling, and then I think I'm going to then just kind of jump in and start the discussion around CRISPR, which is what we're here to talk about today. But just wanted to say, Kate, there's some absolutely beautiful work using Drosophila as a model to understand synapses, those connections between neurons and neurons, or neurons and other cell types like muscle. And she's kind of built this really beautiful image of the synaptic connectivity that's really critically important for communication in the nervous system, and also for kind of experience-dependent changes in the efficacy of signaling within across the synapse. I've seen, if you will want to, we won't show these now, but you go and look at some of Kate's papers. She's got these absolutely stunningly gorgeous images of the synapse, and my favorite ion channel, calcium ion channels, the presynaptic nerve termini. I think when I first saw her talk, I was just like blown away by the power of what she's doing and the beauty of that organization at the synapse. Anyway, that's close to my heart and the love of the things that I do in science. But Kate, like you were also, among many accomplishments as a scientist, you were also one of the first adopters of the CRISPR technology. And so I it's really a thrill to be able to talk with you and get your perspective on the technology. What is CRISPR? Why did it take biology, by storm? And we all know, just less than a year ago now, I think, the two early inventors of CRISPR, Emmanuel Charpentier, now Max Planck Unit for Science of Pathogens in Berlin, and Jennifer Doudna at UC Berkeley, they shared the Nobel Prize for their discoveries. And so it's a big deal. So can you draw us into the world of CRISPR? And
Yeah, happy to. We were really, me and people in my lab and collaborators. So Jill Wildonger, Melissa Harrison, and really driven by Scott Gratz, a graduate student at the time in my lab. And we were really fortunate to actually be working on this question at the time that CRISPR came out. And so we were able to really take advantage of the new technology quickly and adopt it for flies. So it was pretty exciting to be part of, and a really fun time in science. And I think, continuing to have just huge impacts on the way neurogeneticists in particular approach science. So what is CRISPR? So CRISPR is actually a bacterial immune system. And so the way that it works is that bacteria kill invading viruses by cutting their DNA. And so to do that, the bacteria needs to recognize viral DNA. And so that's part of what makes CRISPR so special and why it was borrowed from bacteria for use in other organisms and including humans to edit the genome. So it can recognize specific sequences of DNA and then cut that DNA. And so to understand why that's so important for gene editing, we can think about a document editing analogy. So you're working on your Word document and you notice, it's time to edit, and you notice that you've misspelled a couple words. If you want to go correct those words, you need to get the cursor into the spot where you've made a mistake. And then once you've got the cursor in, you can start deleting letters, adding letters, changing letters. And actually, gene editing works exactly the same way. We need to get a foothold in the DNA. And then once we've broken into the DNA at the specific spot that we want to edit, we can start changing the DNA letters, the DNA code. And so this step, how to break into a specific spot in the genome has been a huge bottleneck for a very long time. And it's this problem that CRISPR solved so elegantly and simply. So it's a monumental task. It's a big ask. The human genome is about 3 billion base pairs. So finding a specific sequence amongst all those ATCGs is not easy. Yet with CRISPR, it actually is. And so with CRISPR, we can readily program the enzymatic system to target specific sequences in the DNA. And it's super easy to program. I mean, I'd almost go so far as to say it's trivial to program. It costs maybe $20 in a few days, and any molecular biology lab can do it. And so this really, really changed the world of gene editing. So CRISPR is a fabulous cursor that we can program to drop in anywhere in the genome and edit. But what it's not is it's not a writer. So it doesn't actually edit the genome itself. So it makes that break, and it opens a window of opportunity for gene editing. And so that window of opportunity is that the break needs to be repaired. So once you've broken DNA, the cell's machinery takes over. And because we face insults like UV light that break DNA all the time, and because that's toxic to cells, we've pretty much all organisms have evolved a number of methods for repairing DNA. And so then those kick in and they repair the DNA. And the goal is to have that DNA repaired, so that you now spell the word correctly.
So the DNA breaks. But the value or the great breakthrough here is that the break is occurring in a highly specific region of the genome.
That's right. So this triggers the opportunity, and it triggers it in the right place at the right time, under your control.
And what I'm hearing you say though is that breaking, okay. But if then you want to go in and actually do very high precision editing in that locus, that's where there's a degree of non-specificity, or at least it's hard to control the specificity of the change that then you want to make. And so can you give us a couple of examples? And I mean, I think my mind goes to, we could think of basic research where this would be really great. Can also think of kind of medical applications immediately. It's been a lot in the news recently.
Yeah, sure. So I mean, so we can do all sorts of things following CRISPR. We can make deletions. So in a lab, you might want to make a deletion because, and we do this all the time, you want to delete a gene so you can understand what that gene does. The genetic approach is to remove a gene, and then you see what goes wrong, and that gives you really strong evidence of what that gene normally does. So we do that all the time. You might want to delete extra sequences that are disrupting a human gene and causing some sort of disorder. And so that happens. I think many of us know about repeat expansions, where you have that underlie diseases where you have a repeating sequence, and sometimes those grow larger, like in Huntington's disease. And when that happens, you could go in and make it smaller. delete some of that sequence. So the other thing that we can do is we can insert sequences. So you could be missing a base pair that then, if you think about a sentence and you think about the spacing of a sentence, and if you maintained the spacing, but you removed a single letter, then everything that follows that is going to not be words. It's not going to make sense. And so that happens with some frequency in genetic disorders. And so those, you could put that letter back. And then we could actually change things. So if the wrong letter is there, we can change it. And the reason we might want to do that in a scientific setting is to make animal models. So if we want to take the wild type version of the gene and we want to change it to a disease variant, then we can go in and we can rewrite the code. So that I answered what we'd like to do. I didn't answer the part about can we actually do it? Mm-hmm. So the answer is yes, but not always. And so what we can do is we can supply the cell with everything it needs to repair the DNA break the way we want it to repair the break. It doesn't mean the cell is going to choose that pathway. And so the cell has really two main options. There are a whole bunch of variants underneath these options that complicate things, but there are really two variants, two general strategies. One is to just glue the broken pieces back together. So you just stick them back together. This is actually the avenue that most of the clinical work in CRISPR is taking. This is a lot easier. You just make the break and you let the cell repair it. And so you think, well, if the cell repairs it, then what have you done? This is an error-prone process. And so often it results in small deletions or small insertions at the repair site. And that can disrupt a gene, which can be very valuable in a clinical setting. And I can talk about a couple of examples where that's being done. It can disrupt a gene or other sequence. And so that's one pathway. The other pathway is a pathway where the cell actually uses a template, another piece of DNA, and it copies that piece of DNA to bridge across the break. And so in doing that, that's an opportunity to sneak in some extra sequences. And so, in the lab, the extra sequences we sneak in a lot of the times are fluorescent tags. And so then every time that the animal makes that gene, it also makes a fluorescent protein attached to it. And so then the protein essentially has, a big flashing green light on it, and we can follow it throughout the cell and see where it goes.
So that's really helpful. Okay, thank you. We've got the framework here. What I wanted to now do is to kind of move into, thinking about we'll go back to some of the more detailed examples in neuroscience shortly, but what I thought I'd do is to kind of pivot and talk a little bit about the impact of the discovery of the technology first, to give everybody a sense of the speed with which this happened. Although I guess speed of scientific technology and creating vaccines right now is kind of given us a new reference point, but by most standards, the speed with which CRISPR was adopted by the scientific community was pretty extraordinary. I mean, I went and looked, and you're again one of the first adopters in Fly, and I'd love you to talk a little bit about that in a second as well. But I went back and I looked at, look at the scientific literature, which is, to get a sense of like what was the trajectory in terms of, when was CRISPR first? I mean, this is an old technology being used for a particular purpose here, but between 2011, so over the last decade, there have been about 24,000 publications that use the term CRISPR, which is a pretty decent number, given that in the prior decade there was about 200. So the rate of increase in the adoption of this technology has been pretty extraordinary just in a decade. And there are journals that scientific journals that are dedicated just to CRISPR, the use of CRISPR technology. And so it speaks a little bit to the impact, but can you give us a sense, like, leaving the medical application to the side for a minute, like what went, like why was this so incredibly rapid, relative to other methodologies? And give us a sense and a couple of examples of why this just took biology by storm.
Sure. So first of all, I'll say that, it was able, so scientifically, we were all able to move very quickly because we knew a lot about DNA repair. We as a community, not necessarily me at the time I was learning, we knew a lot from other gene editing technologies. So we were able to edit genomes before CRISPR. And so CRISPR didn't make something that was impossible possible. It made something that was extremely challenging and only open to a subset of labs working in a subset of systems, into something that was feasible for almost any lab to do, and that had this clinical potential. And so that was part of it. This was a longstanding goal. People wanted to do this. People were willing to invest a lot, a subset of people. And so there was that. There was also the 30 years or so of work on CRISPR. So I really like the analogy to the COVID vaccine. This didn't come out of the blue, right? This built on a whole bunch of foundational science into using RNAs as vaccines, to figuring out how to make that possible. And so this work built on both those foundations, the foundation in DNA repair and gene editing, and the foundation in CRISPR. It also benefited from the sequencing of so many genomes around, prior to that and around that time. And so, you can't do targeted editing of a genome that you don't know the sequence of. And so part of how CRISPR took off is it's been able to turn a whole bunch of organisms that weren't traditionally model organisms that you generally studied in a lab, because you really couldn't manipulate them the same way you could mice, flies, worms. And so it's made that possible. That's part of those 24,000 papers. And so I think it took off because it was, so I can tell you why we adopted it. When we were working on other approaches, we had a different bacterial system that we were collaborating with Mike Cox and Jill Wildonger at UW-Madison to try to transfer into flies to do exactly this, but it was much more complex. And so as soon as this came out, I think it was crystal clear to anyone who was working to make gene editing more feasible, that this was about as easy as it was going to get, this step. And so it was, it was literally one day. Okay, well, this is great. The system we have working, but we're never going to use it, and neither is anyone else. So, what we need to do is get CRISPR working in flies.
That's great. So, Chris, that almost beautifully dovetailing with this discussion. Chris, did you want to
Sure
jump in here?
Yeah, this is really informative for me. I'm glad you reminded me what those 4 letters were that correspond to those base pair things, because I always have to, like, is a cat something? So a question that came in that feels, yeah, exactly in the flow of where we are. So as one of the first scientists to use CRISPR, what kind of difficulties did you face? What was the learning curve like? Where were the challenges? Yeah.
I have to say, it was almost embarrassingly easy. So we had been struggling with gene editing for probably a year and a half prior to that. And so we had been using some of the traditional techniques in flies. And those actually involved attempts to edit genomes without actually making targeted breaks. And that happens, but it's super, super rare. And so if you don't have a system where you can select for very rare events, then it's really painful. And so we were trying to do that and it failed. And so then we were coming up with other approaches to break DNA reasonably close to where we wanted to be. We could get in the general vicinity. And so in doing that work, I think we had worked out a lot of the kinks. But really, I think the biggest issues for anybody who would like to bring CRISPR into a new model system are, how are you going to get the components in? Okay, and this is actually true for humans. How are you going to get the components in? How are you going to get them into the cells you want to get them into? In our case, we were trying to edit the germline so we could make stocks that, breeding stocks that had the edits in all of their cells and all of their progeny's cells. And so, we knew how to do that from a long history of work in flies. That's not true if you're adopting this for, some, animal that's not been studied in a lab. And so delivery of the reagents is a hard part, how much to deliver, things like that. But honestly, those things made it work better. And when we optimized those things, not optimizing those things, the initial attempts, they were lower frequency, but they occurred.
That's really amazing.
Yeah, I mean, it was pretty exciting, I can tell you. So we were 3 junior faculty members, and we knew that we were, wading into a pretty big, an area that was going to explode. And so we gave ourselves 3 months to make it work, and we were able to do that.
Wow. How long between making it work and having that first It's a Genetics paper, right, come out? How long was that time?
It was very short. So it was weeks probably.
Wow.
Yeah.
With the current publication cycles sometimes being measured in years, it reflects how appreciated what you were doing, and in general the importance was at that time.
Yeah, I think that by then there was really starting to be a lot of excitement about CRISPR.
It also, I think that's right, Chris. Right, that that there's a lot of excitement. A lot of people wanted to take advantage of what the methodology that you were using to adopt this into flies. I wonder if you could say a little bit about the importance of, I know that you, and I've heard not from you because you're too modest, but from a lot of people in the field, that you just were so incredibly open about sharing all of your tools with the community that really also accelerated the adoption. And I didn't want to let that slip by, because I think that it's just a really, really, really important component of of science, but certainly how this particular technology really was accelerated, and the discoveries that came along with it were really catalyzed by that ethos, that sharing mentality.
Yeah, and I have to say, we did prioritize openly sharing not just the reagents, but also the approach. We had a discussion board, so where everyone could talk about questions and tips as everybody was trying this, and that was super helpful. And there were a number of people in the community who were very active in that, and so that was absolutely a priority of ours. What's the point of developing a technology other than to have that technology used? And this was just obviously something that many, many, many labs were going to want to do. This was actually, the scientific community in general. I mean, there are 24,000, is that the number? 24,000 papers, because this was able to be adopted quickly because people shared their reagents and their technology. I think that when Feng Zhang's paper came out showing that this was about 6 months after the Genic et al. Paper from Emmanuel Charpentier's and Jennifer Doudna's lab, showing that it could work in a tube. Then several labs came out and showed that it could work in cells. And we were on in a I guess it was probably a Skype meeting in those days, not a Zoom meeting, within a week, and with Fang, and he was sending DNA. So we were able to move quickly because of the openness of others, and other people tried to publish quickly and get their reagents out, and then we tried to do the same thing, and spent a lot of time. We had workshops and things like that to try to really spread the word. And what you really need if you want to adopt this in your lab is those practical tips, right? Like Chris was asking, where am I maybe going to get hung up? What do I do? And what are the variables to play with and those things?
Yeah, right.
Yeah. When there's a famous, well, not famous, but there's a list of 100 of the most cited papers of all time. And I think it's something like 90 of them are either review articles or methods papers. And of the top 10 most, because people so greatly appreciate and want to make sure they give credit to. I think it's a really good thing. It's an interesting thing that it's really about those people that enable science to go forward, so everyone can ask their questions. It has this multiplicative effect.
Yeah, I mean, I feel like this is some of the most impactful work we've been able to do, right? In that, going when we help other labs to adopt CRISPR, then it's not just our lab that's using it and our results and our findings, but it's many other labs' results and findings. So.
Great. So I'm just going to shift to mention a couple of things that are in the chat right now. Before we kind of move on, but there's a really, I don't know if you can see it, Kate, but I'm just going to mention it for the audience. There's a group of high school students who are listening to this presentation. So hi there.
Ask questions.
Yeah, ask us a question. And so they're doing a project, research project on CRISPR's potential to fix color blindness without requiring subretinal injections for our local science fair. So that's fantastic.
Interesting.
So we'll perhaps move back to that, Kate, but I just wanted to do a shout out to say hi to the high school students out there. We all did our local science fairs. So. There's a question here about, once, and I think this is going to help us move toward thinking about medical applications a little bit, but like once CRISPR becomes more widely understood and implemented more broadly, what do you have, or do you want to discuss and wonder about the ethical considerations of being available as a commodity? So yeah. I'm sure that we could talk for hours on this particular topic. And so an example that's given is, what if our families want to modify their children? So can you start us off on a discussion about some of the things that you think personally about when you think about kind of the ethical considerations when thinking about CRISPR technology and bringing it into human use, which of course it is already, but in a limited way right now.
Right. So I mean, I guess I'd talk about, when I talk about or think about using CRISPR in humans, I think about a few major categories of questions. And so, the first one is, can we do it? Can we do it? Can we do it efficiently? Can we actually achieve these kinds of things? In many cases right now, the answer is no, not yet. In a subset of cases, the answer is yes. And so what are the barriers? Efficiency. So what percentage of cells are actually going to be edited when you expose them to CRISPR reagents? That is improving rapidly and dramatically, and I have no doubt that we'll overcome that barrier shortly, or are already overcoming that barrier. Delivery. delivery of CRISPR components to the cells of interest is a real challenge. So how do you get, and so what are CRISPR components? What do I mean when I say that? I mean the RNA, which is what guides the nucleus. So let me just back up a little. I don't think I exactly explained the system. So I said it was easily programmed and it cuts DNA. So those are actually two components. And so it's a pretty simple system. So there's a nuclease, an enzyme that cuts DNA. And that enzyme also binds to what's called a guide RNA. And that guide RNA can be programmed to recognize specific sequences in DNA. And so with the two together, you get the nuclease going to a specific sequence in the DNA where it will cut it. Okay, but so you have to get both of those things into the cell, and then they have to do their job in the nucleus. And so that, if you imagine, for example, a human brain, that's a task we cannot achieve at this point. And so there's that. Then there's the question of, can we do it safely? And so one thing is, there's only, it's ACTGs. Their sequences repeat themselves, or there are very similar sequences elsewhere in the genome. And can you program the guide RNA so that it cleaves specifically at only the site of interest in the cell? If the answer to that question is no, there's the potential that you could break DNA somewhere else. The cell could repair it in a way that actually introduces a deleterious variant where there was none before. So that is something that we have to think very much about. And then I talked about the repair pathway. The cell may not do what you're hoping it will do. It may do something different. And that's especially true if you want to provide a template for changing the sequence from a disease variant to the wild type sequence. If you provide that template, the cell may say, it's faster and easier, I'm just going to glue this back together. And you may end up with a situation where you've either done nothing effective or potentially even caused more harm. And so that, I think the specificity, I think the efficiency, I think those are things that we're making tremendous progress on. Delivery is more challenging, but people are working very, very, very hard on that. People are also working hard on controlling the downstream pathways, but that one's a little bit more challenging. And I think that one's going to take a little bit more time, and explains why in many cases, well, I think in all cases, the current applications of CRISPR in the clinic involve that pathway where you just glue it back together. That's kind of the easier default pathway. And then I think, the final question then is, should we do it? And, that's where you get into the ethics. And that is a question not for scientists to answer alone. That's a question for society to answer. That's a question that's going to be really challenging. It's a question I think that we should be trying to answer now, well before we have the capacity to make changes that start to get into areas where there will be ethical disagreement over what is and isn't ethical. And so I think those are really important discussions that we as a society should be having. We have to think about all sorts of things. What should we do? What shouldn't we do? Who decides? Do your mother and father get to decide for you, what type of human you're going to be? That, may be something that, many of us would imagine might be better left to the individual than to the parents. There is, then of course the question of, and in particular when we're talking, well, no, that's not true. When we're talking about any use of CRISPR, there is accessibility and equity, and is the technology going to be openly available to all? And that's true whether we're talking about repairing disease-associated variants, or if we're talking about potentially in the distant future, providing your children with traits that you feel will be advantageous.
Yeah, this is such a rich, complicated, I completely agree with you that there are people that actually are trained in ethics and philosophy, and that's exactly who we need to be leaning on.
And historians, and and we just need a broad,
Yeah.
a broad swath of society engaged, and and people from different backgrounds, different countries, different upbringings are going to have different opinions about this. And how do we navigate that? I mean, it's tricky, and we need to be actively doing it now.
Is it being done? Do you know? I
It
mean,
is.
to what
It
degree?
is. Yes, it is. So there's certainly the National Academy of Sciences has, convened a large group. Jennifer Dowden is very much involved in this, and so it is definitely being done. I will say there's always the danger of the science getting ahead of the societal considerations. This is something that we see often, and so I think it's important that we make sure that we stay ahead of the science on the ethics.
Yeah, which unfortunately, I think the history of that is that we almost never do. In cognitive enhancement, as someone who drinks coffee and someone who wears glasses most of the time, and I'm certainly in favor of earbud, like there's a zillion things we do to cognitively enhance so some of these debates come down to the medical risk component, I think, that you were emphasizing. I'm just saying that a lot of it is probably going to hinge on the risk reward and where that falls relative to individual choice. But that's just a guess, not being an ethicist or a philosopher.
Right. And also I think the distribution of the technology to equitably to everybody.
Oh, sure.
But yeah, I mean, and safety, I mean, that's another one that, we need to be cautious about and not get too far ahead. That happened in, in more traditional approaches to gene therapy.
Sure.
And there were, there was a teenager, Jesse Gelsinger, who died in 1999. And
Right.
In addition to, the tragic and unnecessary loss of life, it really slowed, it actually ended up slowing the development of gene therapy.
Yeah, with the viral approaches for delivery and things like that. Yeah, very much. Diane,
I was just going to say, I think that I like that Kate really discussed the importance of a range of opinion and a range of experts. And, we scientists have a particular, a lot of times when you discover something, if you're lucky enough to do that in your lifetime career, you discover everything, but like, does anyone care? And when people care, it's super exciting. But that's part of you. It's part of your whole life. Like, this is what you've been working towards. You're not the person to make other decisions. And so, like, because you're just so, I'm not suggesting, but I think there is this emotional investment in anything like that, and so I really appreciate your point about that it's the whole community that really needs to be engaged in this. And I think that that's right. And I actually think universities have an incredible responsibility here, because it's often that, a lot of the basic science and early discoveries are coming out of academic labs, but I think that there's room here, and potentially a really important space to think about this and to kind of lay the framework for kind of some of these early discoveries and engaging the whole community, ethicists, philosophers, historians, and I think a really important point, and I like that to underscore.
And super important that everybody understand what it can and can't do, what are the risks. Right. And so yeah, I think and I think that there are a lot, there's certainly a lot in the mainstream media that's extremely well done. There's some that sort of hypes it, designer babies, but then there's a lot that really or even when you just get past the headlines, that really does a great job of explaining CRISPR for people.
So, Kay, somewhat connected to a couple of questions that come in, I wonder if you could just discuss a little bit about where, you know, there is safety is less of an issue. So, you know, I know at least I should have known this. You probably know. So I know there's the FDA approved clinical trial going ahead for sickle cell anemia, where, you know, the affected cells are removed from the body. You can do gene editing and then put those cells back. Like that seems, you know, that's very exciting. You can select. So I just wonder if you could just, you know, jump into that that application.
Yeah. Yeah, so you know, there are a couple of things to think about when we think about the application of CRISPR in the clinical setting. And so one is, what type of cells are we editing? And what we were talking about that has the much more significant societal and ethical implications is the germline. And so if you are actually you know, editing either your germline or an embryo at a very early stage, such that all of their cells will be edited and they will pass on those edits. So let's leave that aside. That's not happening yet. What's happening is editing somatic cells, so the rest of the body's cells. And so it's happening in two ways. And so this is kind of the other consideration. Can you remove those cells and then edit them and then put them back? So that's ex vivo. Or do you need to, like, let's say, you know, neurons, you know, get them from within the brain, within the body? And so actually both are happening. And so with the ex vivo applications coming first, so I think it was 2019 when Victoria Gray was the first person who was injected, who was treated, whose cells were treated with CRISPR reagents, and then placed back in her body. So these were you know, were blood cells, hematopoietic stem cells. And so in that case, it's interesting, actually. And so that's a case where beta globin is forms an abnormal shape and it causes sickle cell anemia. There are other variants in that same gene that cause beta thalassemia, which is also a relatively prevalent disorder, hemoglobin disorder, that's also been treated with CRISPR in a similar way. So what's happening in those cases, though, is that they're not repairing the variants. Because that we're really not able to do as reliably yet. And so I told you that it's much easier to break a gene. And so that's actually what they're doing. They're breaking a negative regulator of a fetal form of the globin gene, and then once that is then reactivated, it can take the place. It will substitute for and so that so some really clever approaches that are able to use the technology as it's currently safe to use in humans, and so then the other approach is a form of congenital blindness. And so this is the first in vivo CRISPR. And so this is actually injected into the eye. And the goal is to repair a gene that in photoreceptors isn't processed properly. And so what they needed to do was delete this sequence that was providing incorrect instructions for how to process the gene. And so they actually used 2 guide RNAs and they cut here and here on either side of it and to excise it. And so that one's been started more recently, and we don't know anything yet, or at least I don't, about the results of that. But I think it was I think that currently they're in safety trials, and so far it's looking good. And so I will say that, you know, the risks we mentioned, you know, apply. They are using a very high fidelity form of the enzyme to increase efficiency, to increase specificity of the cleavage. And so, you know, I think are reasonably, you know, hopeful and confident about the specificity of the edits.
Great, thank you.
Yeah, there's an interesting question here about a variety of things. So let me offer it to you. So considering CRISPR is taken from bacteria, is it a sign we should be investing more in basic microbiological research and if possible translational applications? And then on a similar note, you know, there's a paradigm of finding clever things nature already invented and then subverting them to something else. Optogenetics is one such example. There are many, right? That's a lot of it. But how far are we from instead being able to just invent the thing we actually want to do? Which is, maybe it's more of a philosophical question, but it does get to a good place in terms of the thoughts about where the genetic engineering approaches should grow. So.
Right. So the second question is a question I'm not sure I can address intelligently about how close we are. I can tell you that, you know, absolutely borrowing from nature and then learning from nature and then applying that. Absolutely, people are doing that. And, you know, it's not just CRISPR, it's, you know, many things that we use, particularly in the lab to manipulate DNA. You know, the entire DNA, you know, recombinant DNA revolution depended on the ability to, you know, recombine DNA, you know, in a tube in specific ways using enzymes that were taken from microorganisms. And so, yes, these studies are critically important. And we absolutely should be focusing on, you never know. You know, I mean, a bacterial immune system that can remember a virus it was exposed to and then, you know, program itself to kill that virus by cutting its DNA the next time it sees it, and to pass that on to its, through you know, through the generations, is a remarkable biological piece of knowledge in the first place, but then you never know where it's going to be applied. In terms of, you know, engineering better or different enzymes, people are doing that through directed engineering and then also through just mutagenesis screens. So randomly mutagenize and then see, is this better? Is this better? Is this better? It's sort of like COVID is doing in nature. So that's an approach.
Thanks. Connected to my previous comment, there's just a question here, and it's I don't know the answer. I'm sure you do. If people use CRISPR to modify the gut microbiome?
I don't actually know the answer. I wouldn't. I don't know. Yes, I'm sorry. I don't know the answer to that.
Yeah, I don't know either. We'll look it up later. Thanks for the question. So there's a question coming in about, you've talked about using CRISPR to alter existing genes and delete harmful, well, make editing sites within the gene. But is there, have you thought about, so there's a question, it's really a question of like adding, rather than getting rid of something that's pathological, you know, one could also imagine augmenting a biological function. You know, that could also be problematic if one is thinking about, you know, like make ethical considerations of that's not just removing and getting rid of a pathological thing or something you don't like, but you could start to try and augment. And so is there an application for CRISPR in thinking about augmenting the genome in addition to altering and deleting. And I think that the essence of this is really enhancing an existing biological function to get more of something great. but there are certainly ethical considerations around that, and you may not actually get what you think. If you augment something, you may not get what you think you're going to get. Like
that
too much of a good thing in biology is typically
that's right. That's right.
a very bad thing.
Yeah, I mean, you know, calcium channels are great, but you know,
yeah. I didn't go there, but
yeah. So yes, we, you know, even too many synapses is problematic, right? So
absolutely.
Quite, you know, there's the biology. There's the, do we understand the biology well enough to achieve that goal? In a few cases, maybe, you know, probably, but you know, many of the complex traits that people would be most interested in augmenting, we really don't know how to do that. And you know, it's not just one gene. Your gene exists in you know, in relation to all of the other genes that are expressed in your body. It exists in relation to the environment, and you know, it acts in relation to the environment. And there are all these complex interactions that are really hard to predict. You know, it's almost like, you can think of it as an ecosystem. You know, we can think about using CRISPR to get rid of mosquitoes that are disease vectors, that carry malaria, that carry Zika, that carry dengue. And, you know, I think, you know, the major concern is, do we fully understand what that will do to the ecosystem? And is it possible that that'll make room for an even worse pest?
Yeah, in agreement with the gut microbiome question, you know, the fact that we've taken this from bacteria also, you know, how does our interactions, which are really deep with creatures that don't share our DNA, how does altering their DNA potentially impact our neural health in any number of ways that, you know, we were talking about getting it into the brain, but getting it into the body components that actually end up making us happy or making us hungry or making us better, improving our immune system in ways that allow us to function better.
Right.
There's both an opportunity and then unbelievable, as you put it, perfect word, ecological complexity to thinking about what happens if we change those other gene components, even the easy ones, the ones quote unquote easy in the microbiome, right?
Right. I mean, there are short sleepers, right? What if we all, you know, only slept 4 hours a day? Do we know that that would be the only effect? You know, would it, yeah, there are huge effects. And I will say, you know, you made me think of when you talked about that we interact with these bacteria all the time. Many of us have actually been exposed to the CRISPR components through our exposure to bacteria that have them. And so there is an additional risk of an immune response to the CRISPR components. And so that one's fairly readily solved because, you know, about 50% of bacteria have a CRISPR system. And so we have a vast reservoir from evolution to choose from. But that's another clinical consideration.
Oh, that's really interesting.
That is I had not thought about that as as an aspect of it.
Very cool. So Kay, we're remarkably, again, staring down the last 5 minutes of this discussion, but I'm gonna, I think I'm gonna combine 2 questions and then we'll see where we get, and then Chris, you can close us out. But so there's a couple of questions here which I'm gonna merge together. One, you know, thinking about the title of the presentation, the use of CRISPR in neuroscience, and everything is so much harder in the brain. But you know, what are you, as you're thinking forward, and especially, you know, around the neuroscience community and the use of CRISPR in the next few years, especially in terms of medicine, neurological disorders, like where's your mind going? What do you think the future is going to look like? And there's another connected question, which is, you know, can we reverse cellular aging? Like it's a big thing that we're all super caring about with CRISPR, maybe. If you know the targets, it's venturing into John Sedevy's area in the Center for Aging. But you know, that's a big set of, you know, what do you see if you think you looked forward to the future and thinking about application of CRISPR in neuroscience, neurological diseases?
So application of CRISPR in you know, the eye, I think, is, you know, is the place where we have the access right now to repair. The role in research and in understanding neurological disease is huge. We can make disease models readily. We can make informative genetic reagents that allow us to really probe how a gene is functioning in a cell. For me, the thing I'm most excited about is, you know, in the brain, it can be really difficult to see the trees for the forest. So there are, right there, you know, it's a dense environment, and particularly for those of us looking at synapses. And so can we manipulate, monitor, follow molecules and function in very, you know, defined subsets? And so one of the things that we're really focused on is how can we follow endogenous proteins, not in their entire pattern, but within a defined circuit. And so we can do that now with exogenous genes by introducing a transgene, but we'd like to be able to do that by following the actual cell's protein. So I think, you know, advances in research already happening, going to keep happening. Neurons are tricky clinically. They also don't do that precise repair for the most part. They don't do that. They do the non-homologous end joining, the ligating back together.
Thank you. That was great. Well, we're gonna, I don't know, Chris, I was just staring at this first question, and I love that we're kind of circling back. That when you mentioned, you know, the retina, this is where the high school students are. And so I like to think like the early career scientists are the people you actually want to follow, because they're aligning and their vectors are going towards what the future is. And so it's very cool. They actually, one of their questions was, how do we find a good mentor? And I don't know if you want to close us out with a few thoughts about how you find a good research mentor. And I'm just gonna say, I'm gonna let Kate say the last words here, but I just did want to also, before we end, thank Sarah Frazier and John Davenport for helping us behind the scenes. We're heading, this is our last talk for this academic year. Well, this semester, I should say, because the academic year at Brown is going through the summer this year. But just wanted to thank everyone for signing in. But Kate, do you want to close us out by, how did you find a good mentor when you were doing this?
Oh, wow. So, I mean, I think that the goal is to find somebody who is passionate and interested and wants to further your career, and, you know, really wants you to follow the path that you want to follow. And one of the best ways to do that is to just reach out, to build your network, to, you know, to dare to reach out, and not just to the potential mentor, but maybe to other people in their lab and other people who might have experienced them as a mentor. And so I would encourage all of you guys to reach out to some of the people doing this work. Describe what you're doing in detail and what you're interested in. And you know, not everyone will respond, but I think you'll get some enthusiastic responses, and those will be the kind of people who you know, might be good mentors for you.
Thank you.
Awesome. Yeah,
great.
Thanks.
Chris, any last comments?
I learned so much. This is fantastic. And
this was really
well
fun.
Explained. Yeah.
All right.
Really well explained.
So thanks everyone for signing on, for the great questions. They just keep us going, and it's been a real pleasure to talk with Kate. Thank you, Kate.
Thank you.
Take care.
Bye.